Hello guys, welcome to my channel. So as you know, we are studying about the carbohydrates. We studied about monosaccharide, disaccharide. So we'll study further about polysaccharide. But today in this topic, we'll study about the sweet taste that we get and how we get that sweet taste on our tongue. All right. So as we know, sweetness is one of the five basic flavors that humans can taste. Correct? The others are like sour, bitter, salty, and umami. So the sweet taste is detected by the protein receptors in the plasma membrane of gustatory cells in the taste buds on the surface of the tongue. Alright, so I will remove this. Okay, so for example, like I'll just draw a simple diagram. So for example, this is our taste bud and it has its cells. Like for example, in this is a gustatory cell in hatch. Okay. So I will explain what is T1R2, T1R3 and everything. And here is CRD. Alright, so this is our taste bud, for example. That's a taste bud cell, gustatory cell, or and this section is just like a receptor, and this is a protein, GTP protein. That's a binding protein, GTP protein, or it's called a binding protein as well, right? This is a binding protein for the taste if you eat like glucose or everything. And for first, this is like an inactive right now, as it does not have any of the like triggers. So like brazen is one of the trigger, like that's the most sweet substance, or like a glucose, right? Or anything like that. So something is coming, and like when we eat something glucose or chocolates or whatever, so this will first bind to this receptors. There are the two receptors that we are just saying. So these are the receptors of the sweet tasting substances. Like taste bud have many more receptors, and each receptors will differ from the, like some receptor do just sweet tasting, some do sour, some do bitter. So like it's separate. It's not like this only does like the sour as well or the bitter as well with the sweetness too. So all it difference. So all the taste buds with the gustatory cell with the receptors are different, right? So the GTP binding protein in humans has two closely related genes. So these are the two closely related genes. So humans has two closely related genes, that is T1R2 and TR3, right? So this is T1, it's not an I. All right, so we have this sweetness receptors that you can see here with that is outside of the cell, protruding outside, right, with the GTP binding protein that is inactive. So when a molecule with a compatible structure binds to this gustatory receptor cells, that is an extracellular domain, it triggers a series of events in the cell. So when it binds, like it triggers a series of events, including the activation of this GTP binding protein. So for example, if this glucose binds here and protein that is activated. So something like this. Some there are like series of events that happen. So where we'll discuss when we'll study physiology regarding the sweet taste. But like when there is a molecule with a compatible structure that binds to this receptors, that is the we have closely related to receptors T1R2 and T1R3. So it develops or it generates a series of events that includes the activation of this GTP binding protein, and that generates an electrical signal to the brain that is interrupted as sweet. So this is how we know that the substance we are eating that is sweet. So like it attached to the receptors, it activates our protein, right? This is activated protein and it generates an electrical signal that is traveled to the brain, and that signal is interrupted as sweet. During evolution, there has probably been selection of uh, ability to taste compounds found in foods containing important nutrients such as carbohydrates that are major fuel for most organisms. So the most simple sugars including sucrose, glucose and fructose taste sweet, correct? But there are other classes of compounds that also bind the sweet receptors. But there are, beside the glucose, fructose and sucrose, there are other compounds as well that can bind to this sweet receptors. So that are amino acid glycine, alanine and serine, right? Other compounds are amino acids, glycine, serine, right? These are also are mildly sweet. These are mildly sweet and these are harmless as well. It does not cause any harm. So these are harmless, right? So this there are other compounds beside the sucrose, fructose. So there are amino acids that can bind to this sweet receptors. Is glycine, serine, there is alanine, and these are mildly sweet and harmless. There is nitrobenzene and ethylene glycol have a strong sweet taste but are toxic. So there are toxic substances as well, like toxic substance that can bind to your sweet receptors are that can bind to the sweet receptors are nitrobenzene and ethylene glycol. So these are toxic substance. Like these are harmless. The glycine, serine, align, right? And these are toxic substances are nitrobenzene and ethylene glycol that can also bind to your sweet receptors. They are like these are mildly sweet and these are strongly sweet in taste, but they are very toxic. There are several natural products as well that are extraordinarily sweet. So for example, stevosides, for example steviocyte. So this is a natural product, right? That is a sugar derivative isolated from a leaves of stevia plant. So these are isolated from leaves of stevia plant, right? Stevia, it's botanical name is Stevia rhabdonia bartoni. This is several hundred times sweeter than an equivalent amount of sucrose, that is our table sugar. The small 54 amino acid protein, brazine, that I wrote here, brazine. And this is a small 54 amino acid protein, small 54 amino acid. Small 54 amino acid protein. So this protein brazing is isolated from the berries of Ubli wine, sorry, in Gabon and Cameroon is 17,000 times sweeter than sucrose on a molar basis. So presumably the sweet taste of berries encourage their consumption by animals that then disperse the seeds so that the new plants are established. So there is a great interest in the development of this artificial sweeteners because of the weight reduction aid. So compounds that give food a sweet taste without adding the calories found in the sugar, correct? So the artificial sweeteners aspartame that we all know, artificial sweetener aspartame, right, demonstrate the importance of stereochemistry in biology. How? Alright, so according to one simple model of sweetness receptor binding, so binding involves three sites on the receptor. For example, bindings involve three sites of the receptor. So one is AH plus, one is B minus, and one is just and X. Right? So the site AH plus contains a group that is an alcohol or an amine, so that can hydrogen bond with a group with a partial negative charge, such as carbonyl oxygen, on the sweetener molecule. Right, so the carboxylic acid of aspartame contains such an oxygen, so they can bind to this AH plus. Right, site B negative, a group with a partially negative oxygen available to a hydrogen bond with a partially positive atom on the sweetener molecule, right? So they also can bond, such as an amine group of the aspartame. So this is how the B negative and the amine group of aspartame can bond together in AH plus, the carboxylic acid of aspartame that can bind with the AH plus, and the same, the site X is oriented perpendicular to other two groups and is capable of interacting with the hydrophobic patch on the sweeter molecule, such as benzene ring of aspartame. So all these three different sites actually can bind with the aspartame on the receptor. So the AH plus contains what it contains. This contains an alcohol or an amine, so that can hydrogen bond. So like this can bond with the hydrogen with a group with a partial negative charge. What kind of group it should be? It should be a carbonyl oxygen. So on the sweetener, it has a carboxylic acid of the aspartame. So that why it bind with the AH plus. So same as the B negative, it contains a group with a partially negative oxygen available to hydrogen bond with a partially positive atom on the sweetener molecule. So it has what it has. It has an amine group of aspartame, and the site X is perpendicular to the other two groups and is capable of interacting with a hydrophobic patch on the sweetener molecule, and such as the benzene ring of aspartame. So when the steric match is correct, when all these three matches are correct, the sweet receptor is stimulated and the signal sweet is conducted to the brain. But when the match is not correct, so for example, if this AH is not matching with the sweetener molecule or B or X, right, the sweet receptor is not stimulated. The sweet receptor will not stimulate, right? In fact, in this case, another receptor for bitterness is stimulated by the wrong stereoisomer of aspartame, and that's why stereoisomerism really matters, right? And the signal will be like it's not sweet, it is bitter, right? And you can see the model that we just discussed. So in the lab it tastes sweet, so how it can match to this three AH plus, B negative, and X, and the on the right you can see the aspartame that cannot match. So this is the same stereoisomer, and there is a two isomers of the aspartame. Alright guys, so this is just extra topic for the sweetness of the taste buds, like how our taste bud detect sweet, what the artificial sweeteners do, and how it can attach to our taste bud and can really brings the signal to our brain. Alright guys, so that was just a small topic regarding the sweetness. Okay, alright guys, so we'll see you in the next video. We'll start with the polysaccharides and we did finish monosaccharide and disaccharide. So we'll start with the polysaccharides. See you in the next video. Alright, thank you guys for watching and don't forget to subscribe to the channel. Thank you.